Circulation Part 3, Blood. Blood has three main functions: transportation, protection, and regulation. Blood is a connective tissue. It is the only fluid connective tissue in our body. It is composed of two main portions: our formed elements, which are going to be our cells, which is our red blood cells, white blood cells, and platelets, and plasma, which is the non-living matrix. Oxygen-rich blood is oxyhemoglobin, and it's going to be scarlet red. Oxygen-poor blood is going to be hemoglobin, and it's a duller red, but you notice both of them are red. It must maintain a pH balance of about 7.35 to 7.45, so just a little above neutral in the basic range. And it has a temperature of about 100.4, which is higher than our 98.6 degrees. When we separate the blood, when we swirl it down and the cells fall to the bottom and the liquid stays up on top, we have two main portions: our plasma and our formed elements. The plasma contains mostly water, some electrolytes, our plasma proteins, albumin, fibrinogen, and globulins, and other substances that are transported by the blood, such as nutrients, waste products, and respiratory gases. Our formed elements, or our cells, are going to be our erythrocytes, which are our red blood cells, our leukocytes, which are our white blood cells that help in the immune defense, and our platelets for clotting. With our red blood cells, they are actually known as anucleated, which means they do not have a nucleus. They have a biconcave disc, which increases their surface area to carry more oxygen. They carry oxygen to all parts of the body. And hemoglobin is an iron-rich protein that binds to the oxygen, and no ATP is required for this. Red blood cells are unable to grow, reproduce, or produce proteins because there's no nucleus. So they wear out within about 100 to 120 days. Once they are worn out, we have phagocytes in the spleen and liver that actually eat the damaged red blood cells and recycle the iron in the liver. Remember, phagocytes use phagocytosis, which is to eat or bring in the damaged red blood cells to be able to recycle that iron. Our leukocytes, or white blood cells, are amoeboid cells, which means they don't really have a certain shape, created in the spleen and liver. They are responsible for our immune defense, protecting us from all the different germs and viruses and things around us. These are nucleated and increase when a foreign substance enters our body. An abnormal increase is known as leukemia, or white blood cell cancer. An abnormal decrease in the white blood cells is leukopenia. We have 5 basic types of leukocytes. Never let monkeys eat bananas. Neutrophils. They help fight against bacterial infections and permeate through capillary walls into the nearby tissues. We have lymphocytes, B and T cell lymphocytes. B lymphocytes help in viral and bacterial infections and produce antibodies. And T lymphocytes fight viral, fungal, parasitic infections, cancer, and transplants. Monocytes, or macrophages, fight chronic bacterial infections. Remember, macrophages use that phagocytosis to eat and engulf those bacteria that they find. Eosinophils play a role in allergies and destroying antigens. And basophils are seen in allergic reactions. They release histamines, causing your blood vessels to dilate and cause more mucus to be formed, which causes an allergic reaction. Here we see that platelets are very small compared to our erythrocytes, but lymphocytes are bigger than our erythrocytes. Here we see a lymphocyte here, and we see neutrophils, which are one of our white blood cells. Here. Red blood cells are smaller and have that concave look, versus our white blood cells are amoeboid, sort of can spread out and don't have a natural shape. We have about 1,000 red blood cells to every 1 white blood cell found in our blood. Plasma is the non-living portion of our blood responsible for the drip. It contains proteins, minerals, sugars responsible for cell repair, and it has 3 main proteins that you need to know about. Albumin, which regulates the osmotic pressure or the water pressure. Fibrinogen, which aids in clotting, blood clotting, and globulin, that are antibodies from B lymphocytes. True or false? All species on Earth have red blood. Well, if we're comparing whether it's red or blue, we know that red is always the correct way to go. Why is the blood red? It is due to the hemoglobin that is found on the red blood cells. Heme is a non-protein pigment composed of iron and a polypeptide chain. The iron helps give that redness to it, and globulin is going to be a polypeptide chain. So again, it's the hemoglobin on the red blood cells that give it its red color. Blood is never blue. Can a person with AB positive blood give to a person who is A negative? Well, we'll hopefully be able to answer that after this next section. Types of blood. We have 4 types of blood. A, B, AB, and O. We can look at it in 2 different ways. Our genotype, which is what is found in our genes, we always show as having 2 different pieces, or 2 letters, because you get 1 letter from mom and 1 letter from dad. That's why you always see these having 2 eyes of some sort. The phenotype is what you see, the physical features. So phenotype is the type of blood. In the genotype, we have a big I that is dominant. So if that is present, it will overshadow the little I. If we have a big IA, that means we have an A protein on the red blood cell. Big IB means we have a B protein on the red blood cell. If we have both big IA and big IB, then we have both A and B proteins on the red blood cells. The little I is recessive. And when we have 2 little I's expressed together, that means we have type O, which means there's no proteins on the red blood cells. Now you're probably wondering, where does that plus and minus come in? We have 1 more protein that can be found on our red blood cells, and that's called the Rh factor, or the rhesus factor. And if it's positive, means we have the Rh factor on our blood. If it's negative, that means you don't have it on the blood. We really won't be using the Rh factor in our Punnett squares, but you might hear it come up later on in this unit. Punnett squares are used to predict a child's blood type, 1 single child's blood type. We say that we use the genotypes, which are the letters, and we say that mother here is type O, so we know that she has a little I and a little I. We use a 4-box square, and we put 1 allele, or 1 of those letters, on top of 1 box, and the other on top of the other box. Now for the father, this father is type AB. So we know the genotype is going to be big IA, big IB. We put 1 letter per row. Then we bring them down, down, and across, and across. The inside genotypes are the prediction of what the child could be. The single child, not 4 children, but what 1 child could be. So in this case, their child could either be heterozygous A, hetero meaning different, because here we have a big IA and a little I, which is different, or a heterozygous B, which again, heterozygous is different. Over here we have a mom that is AB, and a father that is heterozygous B. The other term would be homozygous, which means homo is the same. So that would be, for a homozygous would be big IB, but since he's heterozygous, different, we have big IB, little I. When we bring them down and across, the possibilities for their child would be 25% type AB, 25% heterozygous A, or 50% type B, because both of these could be type B. Just one is homozygous type B and one is heterozygous type B. Some more examples. A homozygous mother. Well, homo is going to mean the same, and she's type A, so that means we have a big IA and a big IA. And the father is type O, which is little I, little I. Means their child is going to be 100% heterozygous type A. That's big IA, little I. A heterozygous mother, big IA, little I, with an O father, little I, means that their child could be 50% heterozygous A or 50% O. Take a look at some visual representations. Type A blood will have the A protein. Type B blood will have the B protein. Type AB blood will have both proteins shown on the blood cell. And type O will have no proteins on it whatsoever. So looking at type AB, this person is used to seeing type B, or protein B. This person is used to seeing protein A. So he's going to be the universal recipient, because he can receive type A blood because his body recognizes the A from having the A on his blood cells. He can receive type B because his body recognizes the type B. He can receive type AB because he is type A and type B. Or he can receive type O because the body doesn't know any different because there's no proteins to say different on type O. So again, type AB is the universal recipient. Type B only has B on its blood cell. So if it receives type A, that's going to be foreign, that's going to be different, and that will cause a reaction, typically a systemic reaction, and death within minutes. So, in the plasma, or the liquid portion of the blood, we have anti-A, which are going to be looking for A proteins. Make sure that there's no A protein. So a B person can receive type B because they're used to seeing B proteins, or type O because O doesn't have any proteins. The opposite for A is used to seeing A proteins but not B proteins. So again, if B is introduced, that's going to cause a reaction. They can receive A and they can receive O. O has no proteins on the surface. Therefore, it can only receive O, because if it receives A, it's not used to seeing that, so that's going to cause a reaction. If it receives B, that's going to cause a reaction. AB will definitely cause a reaction. So they can only receive O. But these are known as the universal donors, because they can donate to every other blood type. So the universal recipients, AB can receive anybody's blood, and O is the universal donor. So here's a quick review that you can go through. Make sure that you can answer all of these because you'll see them on your Open Notes quiz. Two vampires walk into a bar and call for the bartender. "I'll have a glass of blood, please," said one. "And I'll have a glass of plasma," said the other. "Okay," said the bartender, "that'll be one blood and one blood light." That's all, folks.